Hello, everyone. First, I'd like to thank the organizers for the chance to present on my research today, and also James for the lovely introduction. What I've been working on here at UC Berkeley is using a combination of bioinformatics and biochemistry to shine a light on some dark matter in the DNA space, coming from some unexpected sources. That includes gigantic bacterial viruses and this new element, the Borg, which we're still struggling to understand. So first, I started off by taking trillions of these DNA fragments collected from different environments and sort of computationally assembling and studying their DNA sequences. I worked through many data sets from collaborators shown here on the left, but actually the most interesting stuff that I'll talk about today came from my own fieldwork here in California, which just goes to show that everyone has a universe waiting to be explored in their backyard. So first, I got into this world of bacterial viruses or phages from our DNA samples. These tend to be small circular genomes since they rely on their hosts for most aspects related to their reproduction. And one of the biggest surprises that we ended up with were these hundreds of gigantic phages that go from 4 to 10 times the average size of a bacteriophage that we found everywhere that we've looked, and 80% of their genes currently still have unknown functions. Now, you may be wondering, if these are so prevalent, why were they so underrepresented in the literature so far with limited understanding of their roles and what makes their genomes so large? And that's mainly because of inherent sampling bias when people filter for viruses to do this kind of study. So it's sort of like, where's Waldo hiding in plain sight? Once you know it's there, you can find it. And these megaphages, as we've called them, can go up to 735,000 letters of DNA, which is currently the largest phage genome found and exceeds the size of some small parasitic or symbiotic bacteria. And on these gigantic genomes, they encode many genes related to metabolism, but even some ribosomal proteins, translation initiation factors, elongation factors, and an increasing number of tRNAs and translational machinery, the bigger the genomes got. And we saw that they formed phylogenetically distinct clades. So we described and named these 10 new groups of viruses shown on the left here. And one exciting thing that I thought was from these translational machinery, we also saw that these phages had alternative genetic codes where a stop codon was repurposed in some cases to code for an amino acid. And this really improved our gene predictions for those phages where using the right code would give us a full-length gene, for example, here, the DNA polymerase 3, or the RecJ on the right, rather than the fragmented genes we were observing initially. And we recently put out a preprint showing that lysis cassettes are uniquely biased towards recoding. So this suggested that this could be a strategy in the phage life cycle to regulate translation using the phage's translational apparatus. Now, what I'm about to tell you next is another surprise that I thought was one of the coolest findings from these huge phages. So we're all familiar with Crispr systems as cellular defenses against viruses and mobile elements. In bacteria and archaea, this CRISPR array shown here is typically a record of past infections. And you have, in this case, a megasphere cell with CRISPR machinery targeting different viruses within a human subject. But many of these huge phages had their own CRISPR-Cas operons, here with subunits 20 or 30% smaller than in the bacterial counterparts. And we found that many of these huge phages have co-opted CRISPR-Cas machinery to target other competing viruses that in some cases are also targeted by the host machinery to sort of prevail in a superinfection model. And a lot of these systems were much smaller than what was previously described. One system I got very interested in because I exclusively found it in the huge phages that we discovered is the Caspi system. That's about half the size of Cas9, and it still remains the smallest functional CRISPR RNA protein complex, which is important because a big challenge with CRISPR-Cas9 for genome editing or therapeutics is its delivery. And we showed that Caspi can indeed be functional in human cell lines, but also in plants. And another very exciting finding from the biochemistry that we did was that this protein was able to mature its own guide RNA, as opposed to how Cas9 uses a secondary tracer RNA and cellular RNases And not only that, but in Cas5, this is done using the same active site that's used to cut both strands of the DNA target, which in Cas9 is done using separate dedicated domains. So this proved that Cas5 is not only much smaller, but also evolved to be more self-sufficient, so to speak, and more streamlined on the viruses, as opposed to what we've seen in cells. And now I want to shift gears for a minute and talk to you about something totally nuts that we're still trying to figure out in the lab. So within these same samples that we were digging for huge phages in, we found even bigger, more enigmatic elements that we call Borgs. And these replicate in Methanoperidens archaea, a microbe that participates in the oxidation of methane greenhouse gas to the less potent carbon dioxide. Now, these Borgs are linear rather than circular, and named after different colors that we show on the left here, always with two uneven replicors where the genes are encoded on two separate strands pointing in different directions, with about 2,000 bases of long inverted repeats on either side. And there are almost up to 1 million base pairs of DNA. Now we know they can't be part of the host chromosome because the Borg's genome's GC content or GC ratio differs from their hosts, and so do their abundances in the samples, but have only 1 or 2 ribosomal proteins, so they couldn't be a separate lineage or an independent life form that sustains itself. And aligning them to each other showed their relatedness because they seem to have similar blocks of genes, but rearranged differently across different Borgs. And looking at all the encoded genes and comparing that to archaeal viruses, archaeal plasmids, and other extra-chromosomal elements, we see that they actually have more genes in common with their host than with other exochromosomal elements, with very limited similarity to what was previously described. But many more genes also found on the Borgs that are not on the host and vice versa. And we've also seen evidence for acquisition of many genes from the host archaea to the Borgs. And this is really what inspired the Borg name, if you're familiar with Star Trek. One of the gene clusters transferred from the host to the Borgs is this MCR operon that is used for methane oxidation, which hasn't been shown to be horizontally transferred in the past, but also different nitrogen fixation genes and others, but also exchange of genes between various Borgs, which means that multiple Borgs must have coexisted within the same host cell in the past. And another very interesting phenomenon is that all Borgs seem to have these direct tandem repeats throughout the genome, mostly intergenic between different genes, but some also within genes. And the functions of these repeats is still unknown. And looking at what functions are enriched within the Borg genomes, like I said previously, the majority of genes are a mystery with unknown functions, but there are quite a lot of genes for RNA or DNA manipulation, energy metabolism, transmembrane proteins that likely impact the host, and many other functions. And even beyond energy metabolism and electron transfer machinery, there are genes involved in the central carbon pathway, even PHB synthesis, which some may identify as what we call biodegradable plastic in the synthetic biology community, and also in stress response for responding to the environment, and many more. So if there's anything that I would hope you take away from this, it's that there's a whole new world of weird mobile elements out there, full of untapped and uncharacterized genes. And I foresee a future where in my lab, I would be very excited to commit my career to studying such cryptic genetic elements and this genomic dark matter with sort of an eye towards development of useful next generation tools for biotechnology. And with that, I'd like to thank everyone who brought me here and helped me grow as a scientist, all the mentors and mentees, friends and collaborators I've made along the way. And now I'll hand it off to Myrene Balisi from the Natural History Museum of Los Angeles.